In the second part of the lecture, we will discuss label propagation. We'll first show how labeled atlases can be used to segment new images using registration, how we can use multiple atlases, and how label propagation can be combined with deep learning. So first, what is an atlas? Atlas is a single representative subject or a population average that represents a typical morphology or shape and structure of an organ of interest. The atlas also contains the labels that are provided by expert manual annotations. So here you can see an atlas of the neonatal brain. This is the anatomical template. So this is not a single image, but this image has been created by averaging multiple subjects. That's why it's so clear and has relatively simple structure. And this atlas also comes with labels, which is important for label propagation. And here you can see the difference between labeled individual subject and labeled average template. So in individual subject, the cortical folding can be nicely seen, while in the average template, the cortex is much smoother. The reason for that is that cortical folding is individual, and after averaging many subjects, it will become smooth because the individual variation has been averaged out. So for this reason, it might be better to use average template for the registration because it is more likely that we will be able to register the cortical folding correctly, for example. Now, what is label propagation? So in label propagation, we first register the anatomical template to a new subject. And this way we obtain a nonlinear transformation from the template to the subject. And then we can apply this transformation to the labels. And this way we propagate the labels to the space of the new subject. So very simple registration directly will give us segmentation. So this approach has a significant advantage compared to intensity-based approaches because it can transfer clinically relevant information that is chosen by clinicians. And this type of segmentation, these regions in this segmentation do not need to have uniform intensities, and sometimes they don't even need to have clear boundaries. So that way clinician can choose what we will segment rather than relying on intensities of the image to determine what we are able to segment. However, this approach also has drawbacks. The quality of the segmentation is limited by registration accuracy, and registration can also be time consuming. So it can take a lot of computational resources to perform this kind of segmentation. And as an example, registration-based segmentation tend to be particularly poor for delineation of the cortical ribbon. So here, if we compare the label propagation with EM segmentation and to compare the registration of cortical ribbon, we'll zoom in. And here you can see the white matter has been nicely segmented within the cortical folds. So we can see the ribbon nicely delineated. However, in this image where we use label propagation, the details are missing. The white matter segmentation didn't really reach properly within the cortical folds. So here you can see the over-smoothing caused by insufficient quality of the registration. If we have multiple atlases, we can improve the quality of label propagation by label fusion. So what we'll do, we will register all the individual atlases to this to a new subject, and then we transfer all the labels from all the atlases to the space of the subject. Now, to create our final segmentation, we will perform label fusion, and the simplest way to do that is to use maximum voting. So for each voxel, we will count the number of labels for different structures, and we will choose the structure that has the most votes, so by different atlases. So here we can see how the quality of the label propagation improves with the number of atlases. So this is an average dice score of all the structures. And we see that by changing from 1 to 2 atlases, we get the largest improvement. And for a small number of atlases, the dice score still increases. After we reach about 10, the increase is negligible. So even a small number of atlases is useful, as we can see from this graph, and maybe a very large number is not necessary. So the advantage of this approach is that it's robust, because maximum voting increases robustness. And this is because local individual registration failure has less impact on the result, because it might be outvoted by the information from the other atlases. The disadvantage is that we need multiple atlases, which might not be always available. And also it is computationally expensive, because we need to run multiple registration. Now let's come back to the example of a brain MRI of a 2-year-old child. So here you can see single atlas label propagation and multi-atlas label fusion. And just by looking at it, you can see that the single atlas propagated labels are much noisier than from the multiple atlases. So the segmentation is nicely cleaned up. And if we look at it in detail, for example, here we see in multi-atlas label fusion that we have nice connected structures here, thin bits of white matter, and this got disconnected in this image because the segmentation is noisy. So maximum voting is not the only way we can label fusion. We can improve on that. For example, we can select the atlases after the registration by calculating the similarity between the registered atlas and the image. And more similar the image is, more likely the segmentation is going to be correct. So we can either select or weigh the contribution of each atlas to the final segmentation. And this can be even further refined by doing weighted averaging for patches rather than the whole images to get even higher quality label fusion. Alternative strategy is to refine the result of the label propagation based on intensity, for example, using EM segmentation or graph cuts. So let's have a closer look how this intensity refinement works. So instead of label fusion, we can create a subject-specific probabilistic atlas. So let's start with our multiple label atlases. We register all of them to the new subject and propagate these labels, and then we perform averaging of the propagated labels, and you can see the result here. And this can be used as a probabilistic atlas as a prior information for EM segmentation. And the advantage of this probabilistic atlas is that it's subject specific, so it's specifically adapted to this subject. That therefore the prior information will be better quality, and we expect that the EM segmentation will be more accurate. I hope you remember how EM segmentation with probabilistic atlas works from last week's lecture. So let's have a look at the result. So here we see multi-atlas label fusion, and here is EM segmentation with subject-specific atlas, and you can clearly see that we have more detail in the EM segmentation with subject-specific atlas than in the multi-atlas label fusion. And if we zoom in and we here compare the label fusion and two types of EM segmentation, one with population-specific atlas and the other one with subject-specific atlas. So by population-specific atlas, I mean that we create the atlas from the whole population at the beginning, and then this atlas is registered to the subject rather than doing the registrations directly to the space of the new subject. So, as we said, the multi-atlas label fusion results in over-smooth segmentation that lacks the details. So, for example, there is a thin space of CSF that has been deleted. Also, this white matter here and here, and too early, so it's not properly segmented. When we look at EM segmentation, we can see much more detail. But when we compare to EM segmentation with subject-specific atlas, we see that the detail is higher quality. This white matter goes even further into the fold, and here this CSF space is very clearly delineated, while here there are some breaks. So we see that most detail was obtained with EM segmentation with subject-specific atlas. And the final thing on label propagation, I want to tell you, it's the state-of-the-art method called DrawEM that has been used to produce segmentation in the Developing Human Connectome project. So here the atlases actually are a little different. We don't have all the labels in the atlases, but we only use atlases for the brain structures that have some kind of anatomical meaning, but are not necessarily possible to separate based on intensity. So for example, different cortical lobes or different subcortical structures that might have overlapping intensities. So this kind of protocol is decided by the clinician or a neuroscientist to define anatomically meaningful regions. Then, so these atlases are used for a multi-label propagation to propagate it to a new subject. So you can see these structures on the new subject that we want to segment. And the tissues, such as cortical ribbon and white matter, are separated using EM segmentation based on intensity. And once we have these tissue labels that were obtained based on intensity and the structure labels that were obtained based on multi-atlas label fusion, we can merge them to create segmentation in multiple brain regions. So this segmentation is high quality and very detailed. And the final topic for this part of the lecture is how deep learning can be combined with label propagation. So we said that one of the main drawbacks of label propagation is that it's very time consuming. And here deep learning can help because deep learning registration is very fast once it's trained, so we can perform label propagation very quickly. So I will just give you an idea how this works. So this is one of the state-of-the-art registration network that is called voxelmorph. And this architecture of this network is the same as for UNet. But what is different is what is fed as an input and what is an output. So input will be both images, the fixed and moving image. And we want to register the moving image to the fixed. So once we feed the images, then network is asked to predict the deformation field. So this says how we can warp the moving image to the fixed. Now, if we apply this deformation field to the moving image, we get a moved image. So this one is registered to the space of the fixed. And once it's registered, we can calculate the similarity measure. And you already know from previous lectures what these similarity measures can look like. It can be, for example, sum of square errors, or it can be a normal cross correlation or mutual information. So in fact, the loss to train this network is the similarity measure. And the training is fully unsupervised because all we need is the images, and the similarity measure will be our loss and will do all the training. So we don't really need the predicted deformation fields for training. So that's a big advantage. And now once we train this network, we can use it for label propagation. So if we want to do label propagation, we will feed the atlas as the moving image, and the image that we want to segment as fixed image. And the network will predict the deformation field, how to warp the atlas to the new image. And now this deformation field can be applied to the labels from the atlas. So these labels can then be warped to the space of the fixed image, which is our new image, and that way we will obtain the propagated labels. And it will be very quick, and we can do it with multiple atlases as well in a very short time. Now, another application of label propagation combined with deep learning is creating of the training data for deep learning segmentation. So this is actually work from my group done by Dr. Alena Uz that you will see later this term. So what we wanted to do in this project, we wanted to segment individual vessels in fetal heart. And we didn't have any manual segmentations for these individual vessels. So what we did, what Alena actually did, she created an average template of the fetal trunk, which included the heart. And then our clinical collaborator segmented manually all the different vessels. We also have the chambers, but we are not using them for this segmentation network. So then we used registration to propagate the information, the labels from this atlas to a set of images of the individual babies. And we obtained these training segmentations using label propagation. And this training data set was then fed to UNet segmentation network. And here you can see the output segmentations. When you compare this output of UNet with the label propagation, you can see that it's much smoother and better quality. This is because UNet is able to refine the noisy labels from label propagation based on intensity as well. And that way correct the small errors on the boundaries. So we are finding that we get better quality segmentations from UNet compared to the label propagation, which is really nice, on top of being quick once trained. So this is all I wanted to tell you in the second part of the lecture about label propagation. Now please watch the last part of the lecture where we will discuss deep learning segmentation.